I'm Dr. Matthew Davids from Dana-Farber Cancer Institute and Harvard Medical School in Boston, and the co-chair of the Medical Advisory Board for the CLL Society.
Hi, and I'm Bill Wierda from the MD Anderson Cancer Center, and I lead the CLL program at MD Anderson.
Well, it's great to be with you today, Bill. We're going to be discussing in this video an abstract on Pirtobrutinib, which is one of the newer BTK inhibitors. It's called a non-covalent BTK inhibitor because it works a little bit differently from what we call the covalent BTK inhibitors that patients may be familiar with, ibrutinib, acalabrutinib, and zanubrutinib. And we've seen some very exciting clinical data for Pirtobrutinib, even in patients whose CLL has gotten worse on the earlier generation of BTK inhibitors. It suggests that this drug may be effective in that setting. Although it's not yet FDA approved for CLL, we're hopeful that it's moving in that direction. And at this year's ASH meeting, there was a very interesting abstract that was mostly from a laboratory study done at your center, MD Anderson, led by Varsha Gandhi and her group along with you. And this looked at, I believe, sort of what was happening with patients who were having worsening of their CLL while being treated with Pirtobrutinib and trying to explore new combination approaches of how to manage that situation based on the different scientific findings that you had in the lab. So maybe you could summarize for us a little bit about how this study was designed and what it found and what your thoughts are on Pirtobrutinib.
Sure. So maybe I'll start with sort of where we're at with Pirtobrutinib and then move into the abstract that we reported. So Pirtobrutinib, as you said, is a reversible inhibitor of BTK. It binds to BTK and doesn't require the C481 amino acid to bind and inhibit BTK. The C481 amino acid mutation is important because when patients acquire that mutation, in general, it has been associated with resistance to the covalent inhibitors like ibrutinib, acalabrutinib, and zanubrutinib. So at ASH, Anthony Mato updated the Bruton trial data where a number of patients, not just with CLL, but with other hematologic malignancies, have been treated with Pirtobrutinib. And that trial, an update that Anthony presented in the abstract, has shown efficacy and tolerability with an extremely good toxicity profile or lack of toxicity profile with Pirtobrutinib, and activity including in patients who had previously been treated with a covalent or irreversible inhibitor of BTK, and patients who had a C481 mutation. And so the abstract that Anthony presented was, I think, 247 patients who had all been previously treated with ibrutinib or acalabrutinib or zanubrutinib prior to going on Pirtobrutinib. Now, there are some patients who do progress after they've been on Pirtobrutinib. The median progression-free survival for the report that Anthony made was about 19 months. And so about at 19 months, about half of the patients will have progressed on Pirtobrutinib who had been previously treated with a covalent inhibitor and then had gone on Pirtobrutinib. So while there's great activity, it works in patients who are resistant, who may or may not otherwise have a treatment option, but it doesn't last forever. And so there are patients who do develop resistance to Pirtobrutinib, and the abstract that you mentioned is laboratory data that my colleague at MD Anderson, Varsha Gandhi, and her group have generated to try to understand a little bit about the resistance mechanisms and what things we may consider for treatment as the next treatment options for patients who develop resistance to Pirtobrutinib. We have seen, and your group and others have reported, particular mutations associated with BTK in patients who are developing resistance to Pirtobrutinib. So we have also seen that, and Varsha, I think, commented on that in the abstract she reported. But the abstract actually, in the abstract and the work that was done in the abstract, samples from patients who were progressing on Pirtobrutinib were taken to the laboratory and cultured in various or with various drugs, ibrutinib, with AZD, which is an MCL1 inhibitor, with venetoclax, and with combinations of those drugs to try to sort of understand what patients may be sensitive to, at least in the laboratory, so we could think about using those subsequently in the clinic, or at least studying them in the clinic as the next treatment option for patients. And what her data, in essence, demonstrated was that there was sensitivity to drugs in the lab, not to the single agent drugs, but to combinations. So if she co-cultured the cells with the cells that were from patients who were progressing on Pirtobrutinib, if she cultured them in, for example, Ibrutinib plus venetoclax, she could demonstrate that those cells were dying in the laboratory, where we weren't seeing significant death if they were cultured with Ibrutinib by itself or with venetoclax by itself. So this sort of gives us some data to start thinking about next treatment options for those patients. And that's particularly helpful for us in terms of clinical trials and clinical trial options and developing clinical trials for those patients.
That's great. It's really exciting data. I think, you know, the data, the clinical data you mentioned from the Pirtobrutinib trial is also very compelling. You know, we're hopeful that we'll have this as an option eventually for our CLL patients, FDA approved. But I think this story here is also a great example of how, as a field, we always try to stay one step ahead. And I think your group is a great example of that, where, you know, we're hopeful that Pirtobrutinib is going to get approved, but we're already thinking about what do we do after patients have worsening of their disease on Pirtobrutinib? What are the next steps after that? And you know, you got to start with these laboratory studies, and then it can take time to generate the clinical data. So we have to kind of stay ahead of that, so that down the line we'll have additional therapeutic options for patients after Pirtobrutinib.
Yeah, I completely agree. And, you know, we have made remarkable progress in therapeutics for patients with CLL. We still identify high-risk patients who need a new treatment. Those are patients, as I just mentioned, who are developing resistance to the irreversible inhibitors, the reversible inhibitors, patients who have a 17p deletion or mutated TP53. So many of our colleagues think our work is nearly done, but I would not agree with that. I think there's a lot more work that we need to do. And what we are intending to do here at Anderson is to focus on those high-risk patients and see if we can develop curative treatment for them, which also will apply to the patients who have been doing extremely well with the current treatments that we have available.
That's great. It's very impressive and important work. So thank you for doing it, and thank you for summarizing it so nicely for patients today. So thanks, everyone. Take care.
And thank you for inviting me.